Okay. Yes, welcome everyone here in the Bikuben Center and everyone anywhere on the internet. My name is Stefan Bruckner and I will tell you a bit about our virtual poster program that we have this year. Well, first of all, this has been an interesting year for all of us, and when planning this conference, of course, we were faced with several challenges. It was not clear whether we would be able to have in-person part, which we luckily have managed now at all. And of course, this had heavily an impact on how we thought about the poster programs, which we had traditionally and which was always a great opportunity for very lively discussion and a great chance for latest results in progress work, and also for our students to present what they are working on and to create a very stimulating environment for discussion. So faced with this challenge, we said, okay, we have to somehow fix this. We have to somehow provide a poster program. And we came up with the thought, well, we could maybe try to do a virtual poster program. That means that here on this floor, we will not have actual posters present, but the posters are all online, and we have chat room or Discord chat that will be used to facilitate the discussion. So the way that this is going to work is the following. So first of all, I have to, of course, thank the poster committee for their assistance in reviewing all the submission, and we have a total of 15 accepted posters. All the materials for the posters are available on Google Drive, and we also asked all the authors to prepare 90-second preview videos for the posters that we will actually be looking at in a minute. And then afterwards, after watching these preview videos, we will have over the course of the lunch session, but also over the course of the next days, the chance to interact with the individual presenters via text and via voice chat in the Discord. The way that we will do this is that after this fast-forward session, I will actually give you a bit of tutorial on how this will look like, and we are going to do this here live on stage. I'm basically will wing it and see and maybe check in with some of the poster presenters already, and hopefully this will be successful. I don't know. This is not staged at all. So it may in fact end up being hilarious, as Ingrid suggested in the opening session. For now, though, those of you who already have an idea about Discord and how all this stuff works, you may very well already head off into the chat at this link. Those who think that this is some French airplane can still hang on a bit longer, and I will show you how to navigate this. Now, with that, we will be looking at this poster preview video. And again, let me express my great thanks to all the poster authors already for their effort and work in preparing this. Given the whole nature of this program, it's also the case that sometimes with quite short notice, we ask the authors to deliver their materials and to prepare these videos, and they all did this almost completely in time, and with very nice results. So, without further ado, let's look at this video. Also, just as a tip, each poster has a number. You can see those they are presented in order, and you can also see the numbers before each poster, and those numbers match up with the rooms, with the channels in our Discord chat. So that may be an easier way to match up the posters that you're particularly interested in than trying to remember the title, because titles can be long. Okay.
Hello, my name is Krzysztof Krawczyk and I work with a company called Lumito. We are located in Lund in Sweden. Thank you for visiting our poster titled Upconversion Nanoparticles as a Tool for Tissue Evaluation with Multiplexing and Machine Learning Potential. We are developing a novel technology based on upconversion nanoparticles, which have very special properties beneficial for imaging and digital pathology. You can learn more about it on our poster. When comparing our technology with standard DAB-based labeling, you can notice that our images are very clear and we only visualize the marker of interest, while the rest of the tissue remains black. Our technology is fully compatible with digital pathology and delivers clear images with multiplexing and quantification potential. Please visit our digital poster and contact us. We'll be happy to speak with you and answer all of your questions. Thank you.
Hi. In our preclinical project, we look at whole tumor radiomic features extracted from T2-weighted MRI in 24 mice with orthotopic implanted endometrial cancer. We separated the mice in two groups. 11 mice received chemotherapy as treatment, and the other group, 13 mice, received a control injection of saline. We scanned the mice weekly for 5 weeks using T2-weighted MRI. We manually segmented the tumors, and we extracted radiomic features in Syngovia Frontier, and we used lasso statistics to investigate differences between the two groups. We hope you will come and talk to us about our project and poster later today.
What do people coming from the same country and tumors have in common? From the outside, they are often classified as one single unit. However, when taking a closer look, we see that they are actually very heterogeneous, and there are many ways of grouping them. What is a good way to divide tumors in patient-specific substructures? Subregions in multimodal imaging are largely unexplored, so we have to face two major challenges. First, we don't know how many subregions there are, and second, we have no manual segmentation to train with. Therefore, we use hierarchical clustering and pack the results in a visual analytics tool. The user is able to change the number of subregions, and the hierarchical tree resulting from the clustering gives a glance of how regions are split up. Visual exploration, validity measures, and region characterization support the expert during analysis. I am Tanja, and I've been working on this project together with Eric, Ingrid, and Nuska this summer. If you want to know more about the technology, challenges, and future work, we are happy to show and explain you our virtual poster.
Cancer is globally the second leading cause of death. Visual cohort analysis tools can support to develop targeted treatment strategies in cancer. However, the data of cervical cancer patients are gathered over a long period of time and only standardized for the last few years. Thus, building upon the cohort analysis tool by Mert and colleagues, our approach provides users more exploration freedom to find complex relations by enabling double or single-axis dimensionality reduction combined with multiple linked views and group comparison extensions. To see this combination in action, come by our chat room.
Hello, my name is Jakub Jurek, and I'm happy to introduce our experiment. Quantitative imaging, allowing the doctors to measure different properties of tissues, can be thought of as the future of medicine. To characterize a prostate tumor, for example, we would like to know its structural properties like shape or volume, and its physiological properties such as perfusion or diffusion measures. With modern magnetic resonance imaging techniques, we can get insight into both structural and functional properties of tissues. However, do these images have sufficient spatial resolution, which is the key property of image quality? To date, many 3D images are collected as stacks of thick slices, leading to the deterioration of both image quality and the accuracy of extracted measures. In our previous study, we showed how deep learning models trained on simulated structural images can help to reconstruct super-resolution images using information from the acquired low-resolution ones. In our current study, we make the first step to extend that idea to functional imaging by simulating diffusion-weighted images. We achieve that using a diffusion phantom, its computer-aided design model, and MRI signal equations. The resulting images of the phantom are visually compared to the real, acquired ones. Our poster, number 105, describes the details of the simulation pipeline. We invite you to our virtual stand to get encountered. Thank you for attention.
In this project, we have looked at the relation between macro and microscale radiomic features. In the macro scale, we have looked at ADC images generated from mice implanted with orthotopic endometrial cancer. So after manual tumor segmentation, we extracted radiomic features that we have compared to the microscopy images taken from the tumors straight after the mice were sacrificed. So then we look at possible correlations between the radiomic features that were extracted from either the tumor from the microscopy or from the ADC image. So please visit our poster for more discussions about our results.
Mild cognitive impairment, MCI, the intermediate stage between cognitively normal and dementia. MCI is characterized by large heterogeneity, both in terms of brain and clinical outcome. While some patients can live with MCI for several years without progressing to dementia, and some even reverse back to normal, unfortunately, 10 to 15% will develop Alzheimer's disease each year. If we could be able to give an individual disease prognosis, that could lead to better targeting treatment options. In our study, we use high-dimensional longitudinal neurocognitive data from the Alzheimer's Disease Neuroimaging Initiative, the UDNI database. In our poster, we present some preliminary results and also how we plan to implement a hybrid model, the functional random forest, proposed by Feshko and colleagues. Our aim is to identify meaningful subgroups of stable MCI, so we can increase the prediction of further individual trajectories. I hope to see you at my poster.
mild cognitive impairment is a diagnostic construct used to define an intermediate state between normal cognition and different forms of dementias. This is, however, a patient group that has proven to be very heterogeneous, and not all patients will show a trajectory where they go on to develop dementia. It is therefore of great importance to identify predictors of different trajectories within this MCI cohort. This motivated the current study, in which we will use baseline data on test of cognition, global function, and neuropsychiatric symptoms from a total of 754 MCI subjects from the Alzheimer's Disease Neuroimaging Initiative. We will investigate differences between one group that remained stable with an MCI diagnosis and one group that converted to AD over the course of the ADNI study. This poster represents a work in progress, and in further analysis, we attempt to identify and characterize clinical subtypes within this MCI population using a functional random forest machine learning classification model.
Hi, this is the Open Anatomy Explorer, a web-based tool for medical education. Learning about the structure of the human body is an essential skill for almost all medical professionals, but a shortage of donated bodies or even in-person teaching during this pandemic can make this more difficult than it has to be. This is why we have collaborated with the University of Ghent to create a tool to allow them to precisely label their 3D surface scans of real specimens and make them available for students to explore. To facilitate this learning, it is easily accessible on the web and provides tools to create a multitude of easily shared quizzes. This allows the tool to both be used as reference material for students while learning, and also providing a tool to set up spaced repetition exercises and perhaps even for assessment in an exam context one day. There is ongoing work on further extending the projects with features such as more question types, such as orientation questions or freeform questions, as well as linking labeled regions to knowledge bases to make data exploration more comprehensive. In addition, we plan to add volume rendering of CT scans to also display the interior anatomy of the models and provide context to what you're seeing there. Thank you for watching.
Hi, my name is Vilde Brekke, and I'm a PhD candidate at Bergen Center for Brain Plasticity. In this pre-registered study, we combined a concentrated psychological treatment for obsessive compulsive disorder with diffusion tensor imaging, hoping to find how changes in symptoms relate to changes in the brain. Our sample of patients were severely ill before treatment, and yet we were unable to detect any significant differences between the patients and the demographically matched control group. After treatment, almost 80% of the patients were greatly improved and in remission, but we did not find any significant changes in the DTI measures. Our findings cast doubt on how useful diffusion tensor imaging is as a biomarker of treatment response. We are starting a new treatment study with more participants and comprehensive psychological measures, genetics, epigenetics, and imaging to better understand why and how some patients change and others do not after treatment. Thank you.
I'm Hilde Lien, and I'm a PhD candidate in the Bergen Gynecological Cancer Group. In this project, I focus on endometrial cancer samples from patients with a low FIGO stage. Most of these patients have a good prognosis, but some recur. In this dataset of 40 samples, 20 tumors recurred. In order to investigate the different outcome, we use imaging mass cytometry to look at tumor heterogeneity by investigating 27 biomarkers simultaneously. To get the single-cell data, we need to segment the images, and this is done by first classifying the pixel into 3 classes: nuclei, cytoplasm, and background, using a random forest classifier. Probability maps are then generated, which are used in the next segmentation step to create single-cell masks. In this poster, I'm showing a representative analysis of a single tumor sample where we use t-SNE and phenograph to cluster the high-dimensional single-cell data. On the right, phenograph cursors are overlaid on the t-SNE map, and on the left, it is overlaid on the sample image. And we see that there are 6 phenograph clusters that make out 6 distinct cancer cell populations, showing in this single sample we have tumor heterogeneity. To conclude, we have already found distinct cellular heterogeneity with a single sample, and hopefully the entire dataset will tell us even more related to patient outcome.
Hello, my name is Vettler. Digital interaction with volumetric data is increasing in research, the clinic, and industry. In this experiment, we investigated 3D manipulation on 2D screens versus virtual reality. Does stereopsis improve interaction speed? Here I'm demonstrating the task the participants performed. Solving this task requires navigating in a three-dimensional space, either in virtual reality or on a desktop, and understanding the depth structure of the object. If you would like to know more about this project, please come visit us at poster 130.
Hi, my name is Katarzyna Kazimierczak, and today I'll be presenting a project titled Mapping Task and Rest-Related Brain Networks using Topological Data Analysis. This project is intended to look at dynamic interaction of brain activations occurring alongside task-related upregulations. These dynamics are difficult to predict with the current modeling approaches, therefore new tools and methods are required. For that purpose, we have looked at topological data analysis and tried to develop a pipeline which would aid in mapping this dynamical landscape. If you have any further questions, please let me know during poster session, and see you then!
Routine MRI and PET-CT to guide surgical treatment in endometrial cancer. Aim: to describe how MRI and PET-CT findings reported in the standard imaging report have guided endometrial cancer treatment in a prospective population-based 10-year cohort. Study design: we included patients admitted for endometrial cancer treatment from 2009 to 2019 that had undergone preoperative MRI and in most cases a preoperative PET-CT. We performed a patient file review to answer the question how imaging affected the addition of lymphadenectomy to primary surgery. We found that in patients with preoperative high-risk histology, MRI excluding deep myometrial invasion reclassified the tumor from high to intermediate risk in 18 of 50 patients, and in 3 of these, lymphadenectomy was ultimately omitted. For low and intermediate risk patients without additional risk based on imaging, lymphadenectomy rates were 27% and 62% respectively. Of 78 patients with upstaging findings on MRI, 62 underwent lymphadenectomy, and of 22 patients with upstaging findings on PET-CT, 20 underwent lymphadenectomy. Conclusions: preoperative PET-CT and MRI findings help achieve low lymphadenectomy rates in low-risk patients. However, imaging findings indicating reduced risk rarely led to change in the treatment algorithm. Thank you for your attention.
Hi everyone, I'm going to present a poster about multilingual tumor segmentation using bimodal MRI recordings such as T1 weighted and T1 weighted post-gadolinium contrast images. The tumor regions we have considered here are tumor core, whole tumor, and enhancing tumor. Ideally, T1 contrast, T2, and flare images are used to segment brain tumor regions. All 4 MRI recordings may not be suitable for proper co-registration. Also, T2 and flare recordings take longer time to acquire, so we need an efficient model trained on T1 and T1 contrast images only. We have chosen a robust, self-adapting 3D NONI unit built upon vanilla 3D unit the 3D-NN unit is robust over anisotropic imaging data. We have used T1 contrast images and their corresponding marks from brain tumor segmentation challenge 2020 to train our model. It's evident from figure 3 that our 3D-NN unit-based model performs better than the general 3D unit-based model. From figure 2, we can see that our trained model detect two disconnected regions well. The tumor core and then enhancing tumor segmentation performs similar to the model trained on multimodal MRI, but the whole tumor, which includes the edema, provides less dice score. Thank you for your kind attention.
Yes, so I think it's time for already a round of applause for our contributors. Well, and next, let's have a look on how to actually engage with this postdoc program. I had brought a mouse, yes. So you can follow this link to join our Discord server. You can also find this link if you go to the program of the conference at MMIV.no. You can find this here. So what is a Discord server? It's basically if you're into video games or have kids that are into video games, you may have heard that. It actually comes from video games, but also for the maybe older nerds among us, it's basically a copy of something that we had 30 years ago that's called Internet Relay Chat. But for the rest of the people, it's basically an online discussion forum that is also live. So you can interact live, and when you now join a Discord server here, you either can go use a web-based client or as you can see here, or also you can install a desktop application, which can be a bit more convenient. You see, basically looks exactly the same. I'm going to close this one here. And this is what you end up with. Actually, here you can see that, and that's why we used this platform. We actually had a number of conferences this year already that were using this system, so we decided not to reinvent the wheel. So the way that this works now is that in this discussion forum, the discussions are organized into different topics, so-called channels that you see here on the left. And for each poster, there is a text channel. And if you scroll down a bit, there is also a voice channel. And the way that you can use this is you can simply click on text channel. Let's maybe use this one, and in the very beginning of, at the beginning of the channel, you can see a pinned message that I added there that takes you to a Google Drive folder. See here, and this is where the authors uploaded at least the poster and also their preview video. Several contributors have also uploaded additional materials such as video demonstration. So can now simply look at this poster here, say, oh, that looks interesting, but also, yeah, I'm a bit too lazy to read at this moment. So now we could either ask the author a question. So we can go here. And ask them a question, but I saw that we also have someone here. So the way that these voice channels work is, so the text channel, basically, they are always on channels. You can simply switch between the different channels, and the client here will actually tell you whether there are any new messages that you did not see previously. But the voice channels work a bit differently. You see here whether the authors are actually there, whether they are actually present. So it means they are right now in the chat. So when I now join this voice channel, I will hopefully be instantly able to talk to Eric here. Let's see. Hello, Eric. Can you hear me?
Hello, Stefan. Yeah, I can hear you.
You can hear me?
Yes.
Perfect. So, yeah, I was a bit too lazy to read. Can you tell us a bit more about your poster?
Yeah, for sure. So we used hierarchical clustering to figure out subparts of a tumor. I would like to further use them for characterization of the tumor of each patient.
And you have a number of co-authors there. Can you tell us a bit about the background of this collaboration?
Yeah, for sure. So we have multiple co-authors here because we combine medical imaging with visualization. So we are very happy that we have this collaboration here at MMIV. So we work together, for example, with Ink3, and we're very excited to always use medical data as already an application of the tools we are developing.
Do we have maybe some questions from the audience for Eric? No one? Okay, if not, then thank you, Eric, very much for the further info, and the audience here will be able to contact you during our lunch session and in the voice chat. So the authors will be available in the voice chats basically from now over at least until 1. But of course, the nice thing about having a platform such as this is that there are the text channels that you can use throughout the conference and also later to discuss with the authors and converse with them. I just want to show you a few more things and maybe let Eric off the hook here. Maybe. Let Eric off the hook here. If you so here you see the posters, but you can also go. There's another pinned comment that gives you access to this little sheet where you can see all the poster titles. You have direct links to the corresponding Google Drive links with the materials, and you have also direct links to the Discord chat rooms. And you can also rewatch the preview video here. So again, this is available via the links in the Discord, and it's also, I think, linked in the conference program. Yes, I think with that, I don't want to impose my poster preferences on you. I will be certainly communicating with quite a few of the contributors here now, and I suggest you to do the same. And with that, let's finish this session, and I hope you find the poster program interesting.